Remington, if I speak, can you hear me?
Yeah, I can hear you.
Okay, great. Okay, we'll see how many people connect. I hope everyone got the email. Kathy, hi, it's Mike Radovich. Can you leave a sign on the door reminding students that today is a Zoom session? Okay, and then make that for 472 as well. Okay, thank you. Bye. Okay, it's time to start, folks. I'm sorry about this, but standing is very painful. I don't know what I did this morning. I bent over to clean the rubbish out of the drain in the shower. When I stood up, this pain shot through my lower back, and it's been downhill ever since. So walking and standing are very painful. I've not had a back that hurts quite this badly before. So hopefully this will work all right. We'll do what we can. So thank you for your patience. Again, I'm sorry about this, but hey, we don't have to wear masks. So there's an upside. So let's go ahead and, okay. Can everybody see that all right? Yeah, just nod your head if you can. Those of you on camera. All right, yes, good. Okay, if anyone's having a problem, unmute yourself and say so now. All right, looks like it's good. That's where we, well, no, that's not where we stopped. That's where we stopped. Okay, let's start with a word of prayer and then we'll dive in. Father, thank you for this really nice day. Thank you for every student here and their flexibility. Lord, please heal me, because this is a drag. And we pray for this lecture that students would learn a good deal, even though our interaction is not ideal right now. Thank you for this time, in Jesus' name, amen. All right, if anyone did not get the email that today is a Zoom session, maybe just send them a text or something saying lecture's going on, please get on Zoom, blah, 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 so on and so forth. All right, we ended up last time talking about acetylcholine. So acetylcholine has these two different types of receptors. There's a nicotinic receptor. The nicotinic receptor is itself a ligand-gated ion channel. So acetylcholine binds to these nicotinic receptors. They open, they allow cations now to enter the cell. Nicotinic receptors are found in various parts of the brain. They play a role in addiction, and they're also found in skeletal muscle. So these are the main receptors involved with contraction of muscle. We saw this video last time, and then we saw also that the other types of receptors, we'll have more to say about muscarinic receptors, but acetylcholine receptors are regulated. They're regulated by the amount of acetylcholine that they're exposed to because there's an enzyme called acetylcholinesterase, or just cholinesterase. And the cholinesterase degrades acetylcholine. And as you can see here, acetylcholinesterase, otherwise known as just cholinesterase, degrades acetylcholine to choline and acetate. Neither choline nor acetate can bind the receptor, but that limits the amount of acetylcholine that is available for these receptors. And clinically, central nervous system acetylcholinesterase inhibitors are used for dementia, and peripherally, there are others that are used to treat myasthenia gravis, which is a condition whereby the immune system makes antibodies against nicotinic nicotinic acetylcholine receptors. I'm sorry, guys, my room is really cold. Maybe I'm going to turn on my floor heater. That way I'm not having such a problem. There, I'll point it right at me, and hopefully I won't be so blasted cold. Okay, stop sharing. Okay, so let's share this. So here's my makeshift blackboard. So if this, let's say, is a muscle membrane, so say CM, skeletal muscle, and you have here, let's say, an acetylcholine receptor. And this allows the passage of cations, like sodium. You also have out here, let's put these in a different color. And these are cholinesterases. So these are going to take acetylcholine that binds to acetylcholine receptors. So this is a nicotinic, oh, come on, pen. Nicotinic acetylcholine receptor. And this is going to take acetylcholine, and it's going to split it in half to either acetate or to choline, neither of which can bind the receptor. So we'll just say can't bind the receptor. Now, if your immune system makes antibodies, so the immune system is going to make an antibody, and these antibodies are against this receptor. If they bind this receptor, if they bind to this receptor, they're going to cause the cell to take up this receptor and destroy it, or they're going to cause immune cells which have receptors for antibodies in the membrane. And this immune cell is going to damage the muscle cell, and this motor end plate, as it's called, will get destroyed. So this disease is called myasthenia gravis. So asthenia refers to weakness. Myasthenia means it's muscle-based weakness, and it's very severe. So one of the things you could do, since you have fewer of these receptors, is you can use these peripheral cholinesterase inhibitors like neostigmine, drugs like neostigmine and physostigmine, although neostigmine is the one that's used the most. And pyridostigmine is not used quite that much. So these drugs are inhibitors of these drugs inhibit this cholinesterase. Therefore, the reduced number of nicotinic receptors that you have are able to get more function out of the acetylcholine that is made, because there are going to be fewer of them because they're getting removed as a result of this antibody. That disease is called myasthenia gravis. That's another example of drugs that are applied when you don't have enough receptors. Very good. So back to this here. All right, so if a neurotransmitter like acetylcholine binds to a nicotinic receptor and it causes a graded potential in the dendrites and in the cell body, and you are able to get the axon hillock to threshold, that excitatory action potential is called an excitatory postsynaptic potential. The neurotransmitter excites the neuron. It brings the neuron up to its threshold at the hillock and causes an all or nothing action potential at the axon hillock. That is an excitatory postsynaptic potential in EPSP. And these EPSPs caused by acetylcholine, these types of neurons are called cholinergic neurons because they use acetylcholine. Now, acetylcholine can also bind muscarinic receptors. Muscarinic receptors are not ligand-gated ion channels. Instead, they influence ion channels. Instead, these channels are G protein coupled receptors. They're embedded in the membrane. They bind acetylcholine, and then they elicit a response inside a cell that will open another ion channel, in this case, a potassium channel. So here is a G protein coupled receptor. You can see here. Here's your receptor. It binds to the acetylcholine right here. Binds a receptor, and then there is this 3 subunit G protein. This is called the G protein. It has 3 subunits, alpha, beta, gamma subunit. In response to the G protein binding to its receptor, these G proteins dissociate from the receptor, and in this case, the beta and the gamma subunit will associate with secondary targets. In the case of this type of receptor, the beta and the gamma subunit associate with a potassium channel. It binds the potassium channel and it opens it. And now potassium leaves the cell. Potassium now leaves the cell. Now think about this with me. Potassium leaving the cell, is that going to cause the cell to become more or less polarized? It'll cause the cell to have fewer positive charges inside the cell. That will cause the memory potential to be decreased. So this is going to hyperpolarize the cell, isn't it? That's going to make the cell harder to depolarize. This, therefore, is going to be inhibitory. So some of these muscarinic receptors are inhibitory. And they cause an inhibitory postsynaptic potential. Typically because they cause the cell to lose potassium, or maybe they'll cause the cell to gain chloride. Both of those are ways you can hyperpolarize a cell. So this is an example of an inhibitory postsynaptic potential. Inhibitory postsynaptic potentials prevent the neuron from being depolarized. So that's the case with acetylcholine. Some of their receptors elicit excitatory postsynaptic potentials, others inhibitory postsynaptic potentials. A second larger class of neurotransmitters are called monoamines. Monoamines. So these include epinephrine, norepinephrine, and dopamine. Those 3 neurotransmitters are collectively called catecholamines. because they're derived from, or excuse me, chemically, they look like the compound catechol. Then there's serotonin, which is also a monoamine. The catecholamines are synthesized from the amino acid tyrosine. Serotonin is synthesized from the amino acid tryptophan. So monoamines are released from presynaptic vesicles, just like acetylcholine, just like, frankly, all neurotransmitters. They bind to specific receptors in the membrane of the postsynaptic cell. And like acetylcholine, these neurotransmitters are also regulated by degradation. Like the muscarinic receptors, catecholamine receptors are also G protein coupled receptors. Now the alpha subunit of the G protein dissociates from the G protein once the receptor is bound by its ligand, its neurotransmitter. The alpha subunit binds to its secondary target, which in this case is adenylate cyclase. That causes the synthesis of cyclic AMP. Cyclic AMP is what we call a second messenger. Second messenger. And writing with a mouse is harder than it looks. So second messengers like cyclic AMP, like calcium, like other types of signaling molecules, they're made in response to the initial signal. For every molecule of norepinephrine, you make hundreds of cyclic AMP molecules. So this provides a level of amplification, and then this elicits a response inside the cell. And you open cation channels. Those cation channels now allow sodium into the cell. That's going to depolarize the cell, and this will cause EPSPs predominantly. Like acetylcholine, your monoamines are also subject to degradation. So there's two enzymes involved with their degradation. There's monoamine oxidases. Monoamine oxidases, they degrade monoamines. Then there's an enzyme that covalently modifies monoamines called catechol O-methyltransferase, or COMT. Catechol O-methyltransferase adds a methyl group to the monoamine that prevents it from binding to its receptor, and then the methylated catechol is then degraded, but it can't interact with its receptors anymore. So it's prevented from interacting with its receptor and taken out of the picture. And here's a nice picture that shows you essentially the release of the neurotransmitter from the vesicles. This is in response to an action potential coming down the neuron, and then calcium entering the cell, and then you get neurotransmitter here that binds to the receptors, but you also have inactivation by monoamine oxidases and the catechol O-methyltransferase. So serotonin is also called 5-HT. 5-HT stands for 5-hydroxytryptamine. It's used by neurons located along the midline of the brain, the so-called RAFE nuclei. The RAFE nuclei regulate mood, behavior, appetite, and also cerebral circulation. And here is a picture of the location of some of those RAFE nuclei. And the suppression of those are necessary for sleep. Dopamine is used by the dopaminergic system, dopaminergic neurons. They bind to dopamine receptors on postsynaptic membranes. These are highly concentrated, these dopaminergic neurons are highly concentrated in the midbrain of the brain, and their axons project to the nigrostriatal dopamine system. Nigrostriatal dopamine system, dopaminergic system, is involved with voluntary motion, voluntary movement. That's the system that has trouble during Parkinson's disease. And then the mesolimbic dopamine system plays a role in the reward system and is very important for reward and drug addiction and motivation and that sort of thing. So there's a picture from your book, and you see in red, that's your mesolimbic striatal system. It projects to the so-called prefrontal cortex. And then in the blue, up here, you can see the nigrostriatal has its origin in the substantia nigra, which is in the midbrain. It then projects to the so-called basal ganglia in the deep within the brain. So addictive drugs, cocaine, morphine, amphetamines, they activate the dopaminergic system, particularly the mesolimbic system. Cocaine binds to the dopamine reuptake transporter that helps recycle dopamine and prevents it from being recycled. This floods the neurons with dopamine, so you'll need eventually more and more dopamine to elicit the same response. And then nicotine, I don't know if this is on here. Yeah, right here, there's a ventral tegmental area here. That's the origin for the mesolimbic. This then will project to this area here. This is called the nucleus accumbens. Nucleus accumbens plays a crucial role in addiction. Nicotine affects the nucleus accumbens. Neurons of the nucleus accumbens also project to neurons that contain lots of nicotinic acetylcholine receptors, hence the crosstalk between nicotine addiction and the mesolimbic pathway. Norepinephrine, another catecholamine, is used by sympathetic neurons of the peripheral nervous system to regulate smooth muscles, cardiac muscle, and glands. And it's also used in mental arousal. It's used by the so-called reticulate system. The reticulate system is important for wakefulness and arousal and what have you. So amphetamines stimulate noradrenergic neurons that use norepinephrine. Speed, which is an illicit drug, will stimulate these. There are other types of amphetamines you can take over the counter for alertness. These neurons also help with focus, and some drugs like Adderall and then Ritalin, these will help with focus, and they're used to treat attention deficit. Yes, thank you. These are used to extend essentially focus and what have you. Some amino acids act as neurotransmitters. So glutamic acid, aspartic acid, those are examples of excitatory neurotransmitters. One subtype of the glutamate receptor is the NMDA receptor. That plays a crucial role in learning and memory. And it's also a target for psychiatric drugs that are involved with, in some cases, pain, as in the case of ketamine. And it's also used in some cases to treat catatonia and also depression. There's not a lot of evidence, though, that NMDA receptor antagonists actually mitigate depression, but they're still used off-label. So glycine is a inhibitory neurotransmitter. It's used mainly in the peripheral nervous system. It opens chloride channels. It hyperpolarizes postsynaptic cells. And then the poison strychnine blocks glycine receptors and causes asphyxiation because the diaphragm can't relax. And since the diaphragm can't relax, you can't drop the diaphragm down and inhale. Gamma aminobutyric acid is probably the most important inhibitory neurotransmitter in the central nervous system. It's made from the amino acid glutamate. It is an inhibitory neurotransmitter, and the death of GABAergic neurons are responsible for the uncontrolled movements associated with Huntington's chorea. Let me stop here. If you have any questions, go ahead and unmute yourself, or you can use the text function in Zoom. I'm assuming everybody knows how to use that. If there are any questions, go ahead and ask them now. Let me pause a bit, give you some time to catch up on notes. Okay, no questions? All right, then we'll move on. You also have endogenous opioid receptors, and they bind endogenous opioid peptides, endorphin and enkephalin, dynorphin. These are very small peptides made by the brain. They're made to mitigate pain reception, and they are blocked by a drug called naloxone that goes by the trade name Narcan. This is the structure of naloxone. They're made by the central nervous system at certain times. If you have an injury, you start to learn to work through it. So say you lift your leg the first time, it really hurts. You lift it the second time, doesn't hurt quite as much. Something seemingly is taking the edge off it. And that is an example of the endogenous opioid pathways. This is activated by opioid drugs. Then you have gases, nitric oxide, carbon monoxide. These act as neurotransmitters. Carbon monoxide, which is present in very, very low quantities, too low to cause you any damage, are involved in odor adaptation in olfactory neurons. And then nitric oxide, while it's involved in vasodilation, also seems to play a role in the respiratory system and cerebral blood flow and so on, stuff like that. Now, if neurons are receiving both excitatory and inhibitory signals, which one essentially wins, and how does a neuron know which one wins? This is the function of synaptic integration. And synaptic integration is a job played by the cell body. So these two signals will compete with each other. And this comes from your book, and it shows you if you have enough excitatory postsynaptic potentials coming to the cell, you will eventually get the hillock to threshold. And then you'll have an all or nothing potential going down this neuron. And in this case, you may have lots of excitatory inputs coming from several different neurons. And so on and so forth. So that would be an example of spatial summation. Because several PSPs converge on one neuron, it sends the hillock to threshold. If, however, you have one neuron that keeps activating, so you have successive waves over a certain period of time, and that brings the hillock to threshold. That's called temporal summation. So spatial and temporal summation. And this is shown in this picture here, where if you have these two neurons ganging up, if you will, on this neuron, that's spatial summation. And it gets the hillock to threshold. If, however, this solitary neuron here just kept at it, that would be an example of temporal summation. So spatial or temporal summation. Now, if either one of these mechanisms is diminished by an inhibitory postsynaptic potential, then you will not be able to get the hillock to threshold as shown in this slide here. So now one of those, say, neuron number 1 is inhibitory. Neuron number 2 is excitatory. You still have some depolarization, but it's not enough to get the hillock up to threshold. In this case, the postsynaptic neuron will not have enough signal to be able to get the hillock to threshold. You will not have a all or nothing action potential down this axon. This will not occur. Let's just put an X on it here. So the final effect in the postsynaptic cell is more than the input of just one presynaptic cell. Rather, it's the integration of several neurons that converge upon one cell. Now, inhibition can, in fact, be presynaptic. That's the one essentially you see here. But you can also inhibit the postsynaptic neuron itself, or you can actually decrease the amount of neurotransmitter that's released by the axon itself. And this is called synaptic inhibition. Synaptic inhibition. So you can have inhibitory neurons that make a connection with the postsynaptic neuron, and that is going to prevent the amount of neurotransmitter released by that neuron. So that's called an axo-axonic synapse. Here's a picture of it. In this case, you get enough signal. You get an all or nothing potential here at the hillock. But now, this signal, this action potential, will be diminished here. Because this inhibitory neuron is going to decrease the amount of depolarization here, and these voltage-gated calcium channels are not going to give you as much calcium coming into the cell. So your amount of release is going to be pretty wimpy. That's an example of axo-axonic synaptic inhibition. What about the postsynaptic neuron itself? Can you inhibit the postsynaptic neuron itself? Yes, you can. That would be the case with postsynaptic inhibition, like what you see here, and it can be axo-axonic, where the connection is to the cell body, or it can be axodendritic, where the connection is to the dendrites. Both of these will supply an IPSP. So here's an example of an axosomatic or an axodendritic connection, and you can see this here. Here's the axon causing presynaptic inhibition. Here is a, for example, axodendritic. This connection is axosomatic. And here's another one. These are both axodendritic. And they're going to, this is excitatory here. So this would be an axo-axonic connection here. That's presynaptic inhibition here. And this one is causing postsynaptic because you're inhibiting the postsynaptic neuron here. So inhibition can be presynaptic before it gets to the postsynaptic neuron, or inhibition can be the postsynaptic neuron itself. In this case, you can see it's postsynaptic. In this case, you see it's before it gets to the postsynaptic neuron here. It's presynaptic. So inhibition can be pre or postsynaptic. Very good. Are there any questions on any or all of that before we now go to this next set of slides? Neurotransmitters, excuse me, nervous system 2. All right, let's then go to this one here. Okay, I need to share. Okie dokie, let me share you. Share you, okay. And then we'll do that, okay. All righty. Okay. So as we said before, not all of your neurons are alike in terms of their structure. We showed you the different bipolar, multipolar, pseudo-unipolar The brain is full of different neurons like this. It depends on what the neurons do. And the function very often will dictate the structure of the neuron. So this is a slide taken from the book. And that slide, as you can see, is going to show you different types of neurons. These would be multipolar neurons. And then you see the different glial cells, astrocytes, oligodendrocytes, microglia, which are macrophage-like cells that are wandering cells that gobble up cell debris, and then cells that line ventricles. These are called ependymal cells. Ependymal cells. And ependymal cells usually are kind of cuboidal-looking cells. They have cilia on them, as you can see here, and they work to move the cerebrospinal fluid along and help circulate the cerebrospinal fluid. Here's a nice picture of an oligodendrocyte again, reminding you oligodendrocytes make the myelin sheath. If the myelin sheath is damaged because the immune system's attacking it, that is an example of multiple sclerosis. You treat multiple sclerosis by suppressing the immune response. Okay, and this brings us now to the brain. And I need to ask your indulgence, please, at this time, because your professor needs a bathroom break. So give me 1 second, please. This will help you catch up on notes. Okay, I'm back. Thanks, gang, for your patience. All right, so the brain and the spinal cord constitute the CNS. And the CNS, it receives input from sensory neurons. It uses that input to direct the activity of motor neurons. And there are also motor neurons that innervate smooth muscles and glands as well. And that's part of the autonomic nervous system, which is controlled by the central nervous system as well. The brain is also capable of learning and memory, and it's also responsible for consciousness and behavior and so on and so forth. And virtually all these functions map to the brain itself. So the brain begins as a fairly humble tube. you have, here, let me stop sharing. Oh, questions. Presynaptic inhibition connects at the axon, and postsynaptic connects at the cell body or the cell dendrite. It connects, presynaptic connects at the axon of the presynaptic neuron. Postsynaptic connects to the postsynaptic cell. It depends which cell is presynaptic and which cell is postsynaptic. That's essentially what depends on which. That picture that you had previously, I think, explained that pretty well because they had the two cells connected together. If you're inhibiting the cell before, that's presynaptic inhibition, and connection usually to the axon, so it releases less neurotransmitter. That's presynaptic inhibition. And then postsynaptic is going to connect to the dendrite or body of the postsynaptic neuron. That's how I understood it. Okay, and then another question. Okay, thanks. All right. Okay. What was I doing here? Yeah. I was going to do this. Okay. So the nervous system starts out, the embryo starts out as a flat sheet of cells, and there is a little area where cells start to flow into it. This is called Henson's node. Henson's node forms about 14 days after fertilization. Cells will flow in, and then you get this groove. And cells now flow. This then becomes known as a primitive groove. That is about the end of day 14, and it persists till about day 18, 19. This will now extend up this way, and then as it starts to retract, you start to get, if you take this and make a cross-section of it, these cells start to do this. They start to go like this. And then these two ends will join, and they form a tube like this. On the inside of the embryo, like that. So you get this invagination like this, and then you get this tube that forms. So this occurs during the 3rd week. So by the end of the 3rd week, the 21st day, this is well underway. This is called the neural tube. And folks, that's how your brain and spinal cord started, was that humble little tube there, essentially. Here, mousy, mousy. Oh boy. Oh, come on, mouse. There it is. Okay. Okay, now let's go back to you. So the brain forms after gastrulation, and then the neural tube fuses and then goes below the surface, the embryonic ectodermis is called. And so here's essentially kind of the lay of the land at about day, let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. This would be about day, this is about day 19 here. And you can see the tube here has closed. You can see this here. The front and the back are starting to close. There's a hole that will stay at the front and the back called the neuropile. That will remain until about day 21 in the front and about day 22 in the back. But then the brain essentially is going to result because the hydrostatic pressure in the front of the tube is going to cause the front of the brain to expand. And it expands like this. And so third, fourth week, you get this expansion here. That's a prosencephalon or forebrain. This is a mesencephalon or midbrain. This is a rhombencephalon or hindbrain. Then as you get into the fourth, fifth week, starting in the fourth week, getting more mature in the fifth week, these now start to divide. You start to get these little Mickey Mouse ears that grow out. That is the telencephalon. Those are the cerebral hemispheres. They're connected to the diencephalon here. And the diencephalon will form the thalamus and the hypothalamus. And then below that, mesencephalon forms the midbrain and the cerebral aqueduct. Below that is a metencephalon that forms the pons and the cerebellum. And then below that is a myelencephalon, which forms the medulla oblongata. And these spaces are the fourth ventricle here. That's a third ventricle. First and second are lateral ventricles, and then the isthmus down here. And here's a picture of a 28-day-old human embryo, and you can see the two different hemispheres that are formed here. You can see the eye is starting to form here. That's the ear back there. See the stomites. There's the posterior limb bud, anterior limb bud. You can see the liver forming here and the heart. The heart started beating at about day 22, and the heart forms because of inductive signals from the liver, which are forming right here. Inside the structures of the brain are fluid-filled cavities called ventricles. Ventricles are filled with cerebrospinal fluid. The cerebrospinal fluid is a filtrate from blood. It's going to bathe the brain. Transmit nutrients and glucose and things like that to the brain. So the lumen of the telencephalon develops into the lateral ventricles. The lumen of the diencephalon, that's the third ventricle. Space between it is the cerebral aqueduct, otherwise known as the aqueduct of Sylvis. Sorry, that's the interventricular foramen. The aqueduct of Sylvius is the mesencephalic aqueduct down here, or aqueduct of Sylvius. That leads into the fourth ventricle, which is in the pons and the medulla and so on and so forth. This continues to the central canal that goes down all the way through the spinal cord. And if you take a look at it on a side view, you can see the lateral ventricle, third ventricle here, interventricular foramen, aqueduct of Sylvius, and then the fourth ventricle, central canal, so on and so forth. Cerebrospinal fluid comes from a filtrate of blood that's made by the choroid plexus. There are 3 choroid plexi, one in the third and the lateral ventricles, and then one in the fourth ventricle as well. Here's a picture of a choroid plexus. The choroid plexus makes the cerebrospinal fluid, and then the cerebrospinal fluid flows up to the back of the brain here, and they're a structure called arachnoid granulations. The arachnoid granulations, I will spell that for you here. So the arachnoid granulations so they're found at the back of the head. If this is your skull, they're found right back here. There's a little sinus there. The cerebrospinal fluid collects there, and there's these membranes that cover the brain. The arachnoid mater, there's an extension of them here called arachnoid granulations, and they get rid of the cerebrospinal fluid and recirculate it essentially. So it goes near out here back into the circulation. And that's all we have time for today. So if you have any questions, I'll hang out here for just a bit so you can ask them. This recording, I'll put the link on it here, and then the recording itself will be on my YouTube page, but the link will be on our Blackboard page. Okay, thank you for being patient with this. I know it's not ideal, but that's all for today, and I will see you guys. Again, I'll hang out a bit if you have questions. Unmute yourself and ask questions. Thank you very much.